Good afternoon again. Welcome to the second part of the workshop. It's my pleasure now to introduce Jean-François Moreau from the University of Bordeaux. He's been working for a long time at the leukemia inhibitor factor, and he pioneered the work in this direction. In particular, he recognized its importance in transplant. Since then, now he has changed a little bit his interest or expanded, and is working now also at the aging of the immune system. And it's something that is interesting also for physical stimulation. And that's what he will tell us about today. Please, Jean-François, go on. We can't hear you.
You can hear me there?
Yes, now yes.
So thank you very much, Christine, and I would like sincerely to thank you for organizing this very interesting workshop and this gathering. In this short talk, in this short talk, I would like to try to delineate a conceptual frame in which to better understand inflammation. Turning to chronic inflammation means trying to understand inflammation on the mid or long-term temporality. But what definition one can give of the word inflammation? It is beyond the scope, of course, of this talk to really go deep into this important question. Sufficient to say that it has been around for nearly 20 centuries, and there is nowhere in medical practice where inflammation does not make the underpinnings of the pathologies, signs, or symptoms encountered. Asking Google Scholar with the query inflammation retrieve, you see, more than 4 million entries. Today, we are focusing on the use of biophysics to get a better and sharper insight on inflammation. Whatever is the cause of inflammation, inflammation always involves the immune system. The immune system is defined here in metazoans are an array of cells or soluble and soluble molecules, and or also intracellular components are inflammasome that we share with nearly all species in the eukaryotic world, including plants. These components are always mobile and are coordinated over time and space, resulting in the so-called immune response. The immune response first should neutralize the cause which led to the cause of inflammation, and second should organize the repair and restoration to the previous order. The underlying repair mechanism should lead to restoration ad integrum or to different intermediate states of repair, or could never begin, and thus the inflammation state is protracted and becomes chronic without any end. This is the case of many situations, including cancer, aging, autoimmune diseases, cardiovascular diseases, or neurodegeneration. Therefore, inflammation, often considered as negative, could also be and should be considered as a needed phase toward a possible healing, and therefore inescapable. Most of the literature dealing with the immune system and its regulation belongs to cell biology and biochemistry, and much less to biophysics. However, it could be an important missing link to the understanding of inflammation mechanisms and could be an interesting therapeutical target in medicine. The first thing which needs to be emphasized is that the immune responses heavily rely on immune cells' motility and mobility. But what mobility are we talking about? Usually the statement described cells in blood or lymph, as both are mobile fluid in the body. However, recent data have proven that 98% of the lymphocytes, for example, in vertebrates are not in the blood or lymph, but among the cells constituting the tissue or the organ. Could you please start the first video? This short video shot in mouse dermis shows that cells of the immune system, here lymphocytes in green, are heavily represented in skin. Their length of time of residency there could be the entire life organism, and they barely recirculate. Furthermore, night and days, as you can see, they patrol on long ranges and with high velocity, as could be noticed from the scales on this short video. They represent the basis for the local immunosurveillance. Knocking down the cellular mechanism of cell mobility, as it is the case, for example, in DOC8 hereditary insufficiency in newborns, leads to a deep immune deficiency and a strong level of general inflammation consecutive to massive cell death in situ, hereby proving that mobility is a cardinal feature of the immune system. I would like to come back, please, to the slides. Thank you very much. Cells of the immune system are highly mobile and motile among the cells constituting the tissue. As shown in this short video, could you please start this video second? Voilà. Here we follow the travel of one lymphocyte in red among the mouse skin keratinocyte, and which could be seen, they are not mobile. Due to modularity, evolution over the past billion of years, cells lost their mobile capacity inherited from their unicellular ancestors and are tethered between themselves, but also within a 3D matrix called the extracellular matrix. This is this intercellular matrix that constitutes the virtual space where mobile cells of the immune system could circulate more or less freely and perform their functions. Could you please stop it? Thank you. This type of cell migration is also at the heart of a variety of pathophysiological processes such as development, tissue homeostasis, and cancer, to cite a few. The extracellular matrix is a complex entity comprising more than 50 components, roughly half high molecular sugars and half proteins specially organized as a mesh-oriented structure. Collagen are fibrillar glycoproteins, which are among the most represented components of the body and in the ECM also. But non-proteins such as hyaluronans, for example, are also found largely represented. Biochemical composition varies between tissues or among different pathological circumstances. For example, the extent to which collagen fibrils are cross-linked together is highly variable and tend to be specific for a particular tissue. This can be the result of enzymes activity or passive chemical reactions called glycation. In this case, sugars are glucose, fructose, or pentose, depending upon the concentration, may react directly with collagen-lysing residues. This is what is happening in diabetes, for example. This leads to a higher degree of cross-linking, illustrating here the conceptual switch between biochemistry and biophysics. For example, speaking of the immune response, these biochemical alterations modify the stiffness and the porosity of the mesh, hereby heavily constraining mobility of the immune cells and their behaviors in situ. Among many functions, ECM is also a storage medium, creating concentration gradients of soluble mediators or anchoring growth factors to react with cells or guiding their mobility. It also provides, through enzymatic depolymerization of intrinsic components, low molecular weight components called collectively matrikines, playing important roles in inflammation and more generally in tissue homeostasis. Low molecular weight hyaluronans are such an example. It turns out that ECM defined as a specially structured scaffold is pivotal for multicellularity to happen and maintain itself. In this slide is figured a phylogenetic tree of Volvox, which is a sweetwater type of alga made of 50,000 cells. You see this ball of cells, representing in the individual only 10% the cells of this volume, the other 90% being a type of ECM. As can be noticed, Volvox evolved from the unicellular alga called Chlamydomonas as successive multiplication of the initial, say, unicellular pattern. All multicellular intermediate species, tracing back This transition from unicellularity to multicellularity could be found nowadays as fixed species from one cell to 50,000. What is interesting is that for the 10 cell stage on, ECM is found, and knocking down this ECM leads to disappearance of the individual. Therefore, we have to conclude that the ECM is the hallmark of multicellular life organization found everywhere, including plants. Furthermore, it is also found transiently in the unicellular kingdom, as in bacterial biofilms or in the slug of Dictyostelium discoideum, which is a type of amoeba. There are several cellular pathways which link the cell behaviors and fate to biophysical cues enforced by ECM. It includes, among other, ionic channels, motor-actin polymerization, or the YAPTAZ pathway. Collectively called mechanotransduction, these pathways regulate or command many aspects of cell biology, as such as cell size, membrane tension, cytoplasm microorganization, mitochondrial biology, or gene transcription and chromatin organization. In few words, cells are what they are phenotypically thanks to their surrounding ECM and mechanotransduction. One particular aspect of repair in the inflammation context is the extent to which local cells could change their phenotypes and remodel their environment to reach the endpoint of full repair. To give a hint of the consequences of changing biophysical constraints on cell phenotype, I report here a famous experiment where disaggregated epithelial cells from mouse breast are secondarily plated into chemically identical 3D matrices of increasing collagen concentration, leading to increased stiffness of the matrix. At the lower stiffness on the left, disaggregated cells reassemble to create acinar structures reminiscent of the normal breast, whereas at a higher stiffness in the center, the same cells form a tight ball of cells. On the right, increasing further the collagen concentrations and correlatively the matrix stiffness makes the cells become mobile and slowly invade and migrate around. In conclusion, modulating the stiffness of the matrix completely changes the phenotype and the behavior of the epithelial cells. Of course, one crucial question here is where does come from this ECM? The answer is very simple and complicated. Simply, it is made by surrounding fibroblasts, and by complicated mathematical models, it has been suggested that it could be an emergent property of population of fibroblastic cells. This certainly holds true at the beginning of the tissue birth and in the field of development. But once organized, and because ECM is a very long half-life, has a very long half-life, for example, 100 years for collagen found in cartilage, it is susceptible to numerous modificating factors with the consequences one can imagine. One of these factors is certainly the immune system. It is able to infiltrate the matrix and able to change its local organization either via induction of enzymatic cascade or simply by just let the water escape the vascular bed after vascular permeabilization modification, for example. Therefore, it is a two-way, complex and dynamic interplay which is at stake here and which represents an entire new field of biological knowledge to be written and a potent therapeutical target in many medical situations where inflammation is seen. To conclude, I give you an example of a review paper published 4 years ago in Science Translational Medicine that probably many of you know of, speaking of mechanomedicine. It also pinpoints all available means to act on the ECM to alleviate aging, a situation where inflammation is supposed to play a paramount role. The scope of the paper is limited to chemical changes as therapeutical tools, but it cannot be excluded that this endeavor could be reached using other extracorporeal tools belonging to physics, such as pressure, ultrasound, magnetic or field or electric field modification, cold, heat, or light. It therefore appears that the topic of this meeting, once considered a fancy scientific object, is entering in its mature age, and with time would certainly change actively the face of medicine. I thank you very much for your attention.
Thank you very much, Jean-François, for all this. It's really intriguing to listen and understand something more about the interaction between the immune system and mechanical stimuli, and maybe further other type of stimuli. So thank you again. If there's no question, or we can take them later, we can move on to the next speaker. So Professor Carla Stecco, she will also talk about mechanics on a different topic. So mechanical activity, how this modifies fascia, which is a long neglected organ. And her work, her research is mostly turned to understanding at the all levels how this works and how it impacts on well-being in general. She works at the Center for Mechanics of Biological Material at the University of Padua. And please go on with your presentation. Thank you.
Thank you so much for the kind invitation. It's a pleasure to be here with you because I think that we are really looking from different perspectives, the biological tissue and how they answer to the different stimuli. I focused all my research in the fascial tissue from many points of view. The fascia is such a sheet that envelope the muscle, that penetrate between the muscle and inside the muscle, but also in the organ. We have a fascia also in the subcutaneous tissue and support the fat tissue. So it's all the collagen layer that create the shape and the structure of the different organs and muscle. That tissue was neglected for a long time, but now we know that it is very well innerved. It could be cause of pain, the myofascial pain, and above all, it seems that it is very able to answer to different mechanical, chemical, physiological input. So probably it could be a very good target for the therapy. But when we study the fascia, and I think that we need to do that for all the different tissue that we want to target with our therapies, we need to understand what fascia is made of. And in that specific case, fascia is formed by a fibrous component where we have collagen fiber, cells, and the loose connective tissue. And the two elements that both form the fascia have a totally different composition, mechanical behavior, answer to the load, to the movement, to the mechanical input in a totally different way. The fibrous component, for example, the iliotibial band, the thoracolumbar fascia, or other strong fascia, are very similar to tendon, and they are able to transmit the force at a distance. They are formed by collagen fiber, but parallel, very strong. Here we can see the collagen bundle, and we can understand that when we stretch that tissue, the collagen bundle are stretched, are more aligned, and that structure has very few elastic fiber, but is very good to transmit the force at a distance. At the contrary, the loose connective tissue, for example here, is the element that creates the gliding surface that permit to this fibrous layer to glide, to move respect to that fascial layer. Both the elements are important because the fibrous layer creates the support. The loose connective tissue in the middle permit the autonomy, the movement between the different fibrous layer. Here we can see as we can recognize the different element in an ultrasound and also in the histology. But all the different elements could be altered by many different aspects. By aging, by hormones, by diabetes, mechanical inputs, lack of movement. And we are demonstrating one by one all that alteration. I discuss with you only some, above all related with the mechanical inputs. But first of all, we need to understand that we start to have a demonstration that we have a huge changing in the different facial layer according with the aging or with osteoarthritis. For example, in this paper that we have just published, we demonstrate that in the same fascia, the people with arthritis and the people without arthritis, we have a different composition in collagen type 1, collagen type 3, and hyaluronan. So both the fibrotic component and the loose connective tissue in the middle are totally altered. And that creates different mechanical properties of the fascia. The fascia of the arthritic people are much more rigid respect to the fascia of healthy people. But probably they answer also in different way to our input. And here we can see what happened with the immobilization. This is a paper of Slimani, and he demonstrates as the immobilization cause a thickening in the connective tissue. So we have the deposition of a lot of collagen fiber. And in this other picture, we can understand as the immobilization change also the loose connective tissue, the water component, the hyaluronan between the different fascia. And consequently, when we have to treat the patient that have for a long time immobilization, we have a totally different tissue respect to healthy people or what we think that is normal. But also the aging change the mechanical properties of a fascia. For example, in this study in the temporal fascia, they compare a young fascia with old fascia, and you can see that the stiffness of the older fascia is much more. But we know that the fibroblast answer to very different mechanical input. And I think that, at least from a clinical point of view, we need to understand much more how to stimulate the fibroblast to produce more collagen fiber or a better type of collagen fiber, because for a tendon, for a iliotibial band, we need collagen type 1, we need that collagen that is very strong. And above all, it has to be aligned with the mechanical force that act in that specific area. And consequently, it could be for me very important to study if the cyclical stretching could be more effective, how long has to be the static stretching, if the intensity of the stretching, the duration of the stretching, the characteristics of the different elongation to stimulate in the proper way the fascia. And that is just a scheme to understand what happens in unhealthy tissue, where we have all the collagen fiber that have a very precise organization and in a pathological one. When the fascia assumes such a type of organization, totally disarranged, the underlying muscle can't work, and the free nerve endings inside the fascia trigger pain. So we need to work in such type of remodeling to guide the collagen fiber to come back at that direction. But here you can see as the collagen fiber is organized in two different directions, like a network. And is the loose connective tissue, the hyaluronan and the water between the collagen fiber that allow the collagen fiber to move in two different directions. So we have often a double problem, a problem in the orientation of the collagen fiber and in the amount of hyaluronan that form the loose connective tissue and that allow the gliding. So now we can study a little more the hyaluronan. The hyaluronan link the water, the ion in the extracellular matrix and create such type of a web that allow the adaptation of the tissue, that allow the gliding of a muscle respect to another muscle, of an organ respect to the closer organ. But we here we can see, for example, what happened during a muscular contraction when the muscle, more superficial muscle, need to contract respect to the deeper one. We have here a fascial layer that need to glide one respect to the other, the different fascial layer. And the white part are the collagen fiber, but they can move one respect to the other only thanks to the loose connective tissue. And here, oh, I'm sorry, there was a video. Here we can see at the ultrasound the fascia, and unfortunately I'm not able to start with the video. But that is the deep fascia with the three different white layers and the loose connective tissue in black in the middle. And this is a healthy subject, and we can see the three different layers that move one respect to the other. On the contrary, in this other subject, it was evident, I'm sorry that I can't show you, a lack of gliding, a lack of movement between the different tissues, and consequently the underlying muscle can't contract. That lack of gliding was demonstrated for the first time by Professor Langevin of Harvard University in the patient with a specific low back pain. She demonstrated that such type of patient are not able to glide, the erector spine are not able to glide respect to the thoracolumbar fascia. And that is related to a lack of range of motion, the intensity of the pain of the patient. So probably we need all the hyaluronan, the water component, and the viscosity of the hyaluronan can play a key role in the fascial pathology. So Professor Langevin did also a second study in the animal model, analyzing how the restriction of the movement, the surgery can affect such type of gliding, and both can change the gliding. But she also tried to train the animal model with stretching, and she demonstrated that the stretching is not enough to restore the correct movement. So for that reason, I think that probably we need to consider much more the physical therapy. And consequently, I try to study more the hyaluronan, and in this paper, I demonstrate as the different fascia of our body have a different amount of hyaluronan according with the movement of that specific area. The retinaculum, that are the fascia close to the joint, have a much more hyaluronan respect to other fascia that are in area where there is less movement. But for example, in the plantar fascia, we found a huge difference of the amount of hyaluronan into the fascia, suggested that probably there is a variation among the population, and we need to understand much more what happened. But also in the neck, in the patient that have neck pain, we found an alteration of the hyaluronan. And I think that the hyaluronan could be a very important target, because the hyaluronan is sensitive to the pH, and it can change the viscosity according with the pH. We need to remember that after strenuous exercise, we can reach a pH of 6.6, and that can increase a lot the viscosity of the hyaluronan. But the hyaluronan is is affected also by the temperature, and if we warm the tissue, the hyaluronan become more fluid, and consequently the muscle can glide much more. So I think that some physical therapy that work with the temperature can help to improve the gliding of the hyaluronan. And we have repeat such study also with the physiological temperature, and you can see a change of the viscosity with the temperature. And then we use the shock wave, because the shock wave are a sound wave, so they create a direct mechanical stimulus into the fascia. We treat in the lab with the probe in such a way, we create a support with a 3D printer in the some cell culture coming from the fascia. And we demonstrate that under the stimulation of the shock wave, the cell start to produce a bubble of hyaluronan, and that bubble are released in the extracellular matrix. We demonstrate that with the microscopic and the electronic also microscopy. Here you can see the release of the bubble of hyaluronan in the extracellular matrix. And that release starts just after 4 hours. And we maintain that production also after 24 hours. What means that probably one of the effects of the shock wave into the fascia is that they stimulate the cell, the fascial cell, to produce more hyaluronan, and that can increase the gliding of the fascia. That is one of the elements that is altered in many different pathologies. That the work was similar to another result obtained by the Dias dos Santos into the bone. Also him demonstrate increased production of hyaluronan after stimulation, and that stimulation was also for many, many days. At the same time, another paper demonstrates as the shock wave are not able to increase the collagen type 1, the fibronectin, the TGF-beta, the alpha-SMA. So probably it works much more in the hyaluronan rather than in the fibrotic part of the fascia. And we need to be very precise when we consider the effect of the mechanical input in the biological tissue, because the biological tissue has many, many aspects. We have the cell, we have the extracellular matrix, we have the fibrotic component, and the different physical therapy probably work in specific part of that element, and we need to be precise what we really want to do. So to conclude, we can affirm that the fascia surely answer to the temperature, to the pressure, to the mechanical input, to the stretching, to the pH, but all the different input affect the fascia in totally different way. Some work more in the fibrotic component, some work more in the loose connective tissue. In the patient, some patients have more problem of gliding, others have more problem in the fibrotic organization. And consequently, if we start from this perspective, we can organize and understand much better the indication of the different physical therapy. Thank you for your attention.
Thank you very much for this presentation. Indeed, the more we learn, the more it gets complex. So you have some questions here. Oops, sorry, let me read. So from Jean-François Moreau, how are the muscle of nude mole rat organized? They are characterized by an overproduction of hyaluronans. They live long with no cancer.
I never check such a type of fascia. I'm very curious to analyze them. And thank you for the suggestion because I learn immediately something new. And surely could be a nice perspective to analyze such type of fascia.
Okay, then you have a couple of questions from Vitaly Zablotsky. What is the reason to use 2.5 hertz for stimulation? Is there a connection of the 2.5 hertz with a relaxation time?
I'm not a physical expert in the physical properties of the shockwave. I use more in the clinical practice. We tried many different level of energy because it was a problem to understand, to avoid the damage of the cell. These input were done in cell culture, so we had to reduce a lot the energy to give to the cell because at the beginning the cell were destroyed by the shock wave. And reducing the input, we were able to arrive to a level that were able just to stimulate the cell without to create a damage of the cell. We have checked also the vitality of the cell in this work. And that was the level to permit a good vitality of the cell and give at the same time the stimulus.
Thank you. Then from Laura Calza, you mentioned Dr. Langevin, the director of the National Center for Complementary and Integrative Health. Does your research intersect acupoints and meridian anatomy?
Yes, I have a long time collaboration with Professor Langevin. And my research about fascia are the anatomical support of the meridian's anatomy. So that because the fascia are organized in a very precise way, and their anatomy, the reinforcement of the fascia are exactly overlapped with the meridians. So I collaborate also with China and with different acupuncture school in China for that reason. So the energetic concept of the meridians probably have also an anatomical basis into the fascia. And the orientation of the collagen fiber probably permit to a faster communication along a specific line.
Thank you for this. We have a couple more minutes. Do you want to try to show your video by sharing the screen? So if you go to present, share screen, maybe you can show us your video. I think we need to, Biagio, maybe we need to remove now the presentation so that she can upload. Okay. Share screen. Okay.
Are you able to see my screen?
Yes.
Okay, so I can go here.
Maybe you can have a full screen so that we see better. Okay. Thank you.
Here, not allow me to see the video. I have to show you also with the presentation like that, but I show you from another. Maybe if I'm able. No. Don't allow me to show the video,
Okay.
so
Maybe
I don't understand why.
If it's like this, you can try to upload it, and if we have some more minutes later, we will put it on air. So you
Okay,
can
perfect.
upload it as
I can do that, of course.
Yeah. So present video file, you upload it, and then we try to have some time later on. So thank you
Okay,
again.
thank you so much. Thank you.
So now it's my pleasure to introduce Luigi Manni. He's a neurophysiologist also at CNR. He has an expertise on the effect of electrostimulation on the nervous system and a particular interest in the function of the nerve growth factor. So he will tell us about his expertise. Please, Luigi, the floor is yours.
Okay. Thank you, Christine. Good afternoon to everybody. And can you see my presentation here?
Yes, you can
I'm
go.
not. Can you see the
We
presentation?
see. Yes.
Okay, I will go. So in the next minutes, in the next 15 minutes, we will try to take a brief look at some of the mechanisms through which sensory stimulation, especially the stimulation that is transduced by mechanoreceptors, can modulate the inflammatory response. So first of all, I would like to say that when I use the term mechanoreceptor, I indicate the more or less specialized endings of nerve fibers that are responsible for perceiving and transducing stimulation applied to the surface of the body. So I'm not talking about molecules or receptor at the cellular level, but I'm talking about sensory nervous system. A very effective way to activate mechanoreceptor on the surface of the body in a way that is controlled and reproducible is through acupuncture. And of course, you cannot see the video here, and there is a video anyway. I will show probably later of rats experiencing electroacupuncture while they are awake in our lab. So we know that acupuncture is a powerful form of sensory stimulation since the beginning of scientific research in the field. Indeed, in an experiment in the early 70s that, if I remember correctly, was done on healthy volunteers, healthy students at the Berkeley campus of the University of California, it was demonstrated that acupuncture, a single session of acupuncture was able to increase the nociceptive threshold in human volunteers, that the changes in this threshold was gradual and then decrease after the cessation of the stimulus, and this is indicative of the presence of the mediator of a diffusible factors mediator as a mediator of this effect. The discovery of endogenous opioids was more or less dating 10 years later. And most important for our reasoning is that this effect was blocked by procaine, so by a local anesthetic injected at the place of needle insertion. And this was demonstrating that the sensory afferent, the sensory system, was involved in the effects of acupuncture. So when you insert a needle at the body surface and you stimulate the needle, that is something that is compulsory even in traditional medicine, and you can stimulate by manual rotation or by electrical current, for example, you may activate all types of mechanoreceptors present in the dermal layer of the skin. I'm talking about Merkel corpuscle, Ruffini corpuscle, Pacini and Pacini corpuscle, or muscle spindles in the muscle. And by gently and manipulating the needle, by repeating this operation, you are expected to produce a mechanical pressure and tissue distortion that can activate all these kind of receptors. And so These are receptors that differs among others for their, for example, for their adaptation properties, and they are classified into slowly and rapidly adapting. As you can see, the slowly adapting receptor can respond through all the time that the stimulus is applied, while the rapidly adapting sense the beginning and the end of the stimulus, transferring action potential only at the beginning, at the end of the stimulation. The mechanoreceptors also differ for their sensory threshold, that is the intensity required to generate an action potential in the nerve fiber. Usually rapidly adapting receptors have lower thresholds than slowly adapting receptors. And all these receptors also differ because of the fibers that innervate these receptors and the nerve conduction velocity of these fibers, that is proportional to the myelin that is sheathing this fiber. So the large the diameter of the fiber, the higher the velocity of conduction. All these differences can account for the differences that have been reported in receptors activation and in the effect and the outcome and the clinical outcome for different modalities of stimulation. So manual stimulation, electrical stimulation are low or high frequencies, are low or high intensities, can stimulate different receptors and then exert different effects mediated by different endogenous mediator. So in this table are summarized the various mechanoreceptors and the types of nerve fibers that innervate these mechanoreceptors. The range of fibers that innervate the receptor in the dermis goes from slow to medium fast conduction velocities, while the fibers of the receptor in the muscle have higher speed of signal conduction, is the kind of fast type fibers. As I previously mentioned, the receptors are the terminals of the sensory fibers that originate in neurons that are located in the dorsal root ganglia, and through these neurons, the receptors are connected to the spinal cords, to the dorsal root of the spinal cords. The spinal cord is the first part of the central nervous system in which the sensory stimulus is integrated and processed in accordance with the classic scheme of homeostatic regulation. Indeed, acupuncture through the repetitive and controlled stimulation of mechanoreceptor could modulate the activity in the control center, that is the central nervous system, and finally, ultimately affect the effector response, as we will see in next slides. This is there is an animation, but anyway, when you apply skin at the needles of the skin surface, you always have three effects at three levels of the nervous system. A first local level that is mediated by the activation of nerve afferents, is a local level, and the mediator are mediator inflammations such as prostaglandins, serotonin, growth factors, or neuropeptides. Then there is a second level in the nervous system, the spinal segmental level, in which you can have a reflexive modulation of the function of visceral organs that is mediated by the autonomic nervous system, mainly the sympathetic nervous system. And then you can affect the way in which the central nervous system controls, for example, the autonomic activity and the pain descending control system. You can exert a control by acupuncture on the sympathoadrenal and hypothalamus pituitary adrenal or gonadal axis. And finally, possibly you can also affect higher brain functions like mood or emotions, and you can also, again, give a feedback or descending regulation of pain and of autonomic activity. So there are basically two ways in which we can modulate inflammation using acupuncture. The first is based on the capacity of acupuncture to generate the axon reflex. The axon reflex is a response in which impulses initiated in a sensory fibers can be relayed antidromically down other branches of the sensory nerve fibers. And this means that action potential are transmitted antidromically and can generate the release of neuronal mediators such as neuropeptides, CGRP or substance P, that in turn can induce the granulation of mast cells and production and release of mediators in the vessels, and finally induce vasodilation, extravasation, and edema. This kind of reaction, this is the so-called neurogenic inflammation, is thought to be protective and facilitating the process of wound healing, of tissue healing. So, and this is a reaction that is always present when you use needle at the surface of the body. As you can see, the flare reaction and the redness in the point in which the needle has been inserted. We in my group we studied over time the effects of acupuncture on some of the mediators, the local mediators of this kind of response, and namely nerve growth factor and neuropeptides. And so we were able to demonstrate, even though not properly in an inflammatory model, but we were able to demonstrate that we can reduce the insurgence of pain, of neuropathic pain, and at the same time affect the distribution and the expression of NGF receptors in the skin. As you can see here, animals treated with acupuncture are similar to control in comparison to diseased animals for P75 expression in the skin, and also in the DRG, in the neurons of the DRG, where we observed both the reduction of the activation in TRAC receptor and in the presence of P75 receptor. We were also able to decrease, to inhibit in some way the action of NGF on the expression of pain and inflammatory mediators, such as the vanilloid receptor 1, that is ion channel highly expressed in C fibers, in unmyelinated sensory fibers, and in the corresponding neurons in the DRG, and of the neuropeptide substance P. Both of them are controlled in their expression by NGF. And interestingly, we were also able I repeat, not in a proper model of inflammation, but we were able to affect the NGF system and the expression of NGF in this, as reported in this slide, by acting on the reflexive modulation in the spinal segmental effect of acupuncture. Here we decreased the ovarian content of NGF in rats affected by experimental polycystic ovary, by needling the animal in the thoracolumbar section of its body. And interestingly, we obtained the same. This effect was relayed on a mechanism that involved the release of noradrenaline by sympathetic fibers in the ovary and the activation of the beta-2 adrenergic receptor that in turn controlled the NGF production in the ovary. And interestingly, we also observed the same effect when we applied the voluntary exercise as a therapeutic intervention, And this is, of course, a modality in which mechanoreceptors, especially those in the muscle, are really challenged. So there is also another local possible mechanism, that is the one which rely in the release of adenosine phosphates by keratinocytes or mast cells after mechanical stimulation of the epidermal layer, and this has been is an argument that has been treated in previous presentations, so I will not go deep in this matter. The second way in which we could affect inflammation by acupuncture is by acting on the inflammatory reflex. And this effect is based on the capacity of this kind of therapeutic techniques to affect the activity of the vagus nerve by controlling the activity of the central dorsal vagal complex neurons, and also by the capacity of acupuncture, as I mentioned before, to control the sympathetic efferent flux. By acupuncture we are able both to affect the sensing of sensory inflammation that is in charge of mainly of the sensory vagus nerves, sensory vagal afferents that sense the presence of interleukin and of inflammatory cytokines and mediators in the tissues and in the circulation. And we can also modulate the effector response by modulating the release of acetylcholine by vagal efferents and the release of noradrenaline from sympathetic efferents. Both of them have been demonstrated to have an anti-inflammatory effect, acting on pro-inflammatory cells. At the same time, we can control the activity in the hypothalamus pituitary adrenal axis and the release of anti-inflammatory hormones such as ACTH or glucocorticoids. This is so we can control. We can have a control on the neuroregulation of inflammatory response, both on the acting on both diffusible factors and on the nervous activity that are involved in this inflammatory reflex. So in our recent research on the effects of acupuncture on the central nervous system has been focused on basically on the learning and memory functions and associated brain areas that are not properly done on inflammatory models. So I will not bother you with data from my group that are not related to the main topic of this presentation and of this workshop, but I would rather call your attention on data from other groups that, in my opinion, should be considered in some way. They call for two main considerations. First data has been obtained on a model of peritonitis in rats, and researchers compared the effect of pretreatment with manual acupuncture with those of the indomethacin, that is a FANS. And the effects have been measured on the cytokine levels in peritoneal fluid. And as you can see, we have the same effect for acupuncture and for indomethacin, for both in the diminution of pro-inflammatory cytokine and in the increasing of anti-inflammatory mediators such as interleukin-10. And this call for a first consideration, that is maybe we can avoid a lot of side effects induced by this kind of drugs using the anti-inflammatory properties of this kind of physical stimulation, at least in some kind of inflammatory disease. And the second consideration that I would like to do is coming from this data from another group. They measured the the serum TNF after manual acupuncture in mice systemically injected with LPS. And they saw a rapid effect of acupuncture in decreasing the TNF, circulating TNF, an effect that was abolished by or partially abolished both by sympathectomy of the splenectomy, or by vagotomy. And this means that both the effect of the acetylcholine and both the parasympathetic and the sympathetic branches of the inflammatory reflex are active and needed to dampen the inflammation after stimulation with acupuncture. And in the in a later time after stimulation, they see that this effect is decreasing, but this effect of splenectomy or of vagotomy is decreased, while it remains the anti-inflammatory effect of acupuncture. And this probably is due to the diffusible factors effects that is taking place at later time after the stimulation after the needling. And so I finish my presentation. Thank you for your attention.
Thank you very much, Luigi, for this information and this explanation. We are slightly late, so we will try to send the videos, both yours and the one of Professor Stecco, later. So now we exchange role. You moderate.
Yes, of course. So we exchange role because now is the time for us to listen at the presentation of our guest. That is, as you know, is Christine Nardini, PhD in electronic engineering at Alma Mater, actually at the Institute for Applied Computing at CNR in Italy here, after former experiences as a professor at Max Planck Institute and Chinese Academy of Science in Shanghai, and as associate at the Department of Laboratory Medicine and Chemical Chemistry at Karolinska Institute in Sweden. Thank you, Christine, and go on.
Thank you very much, Luigi. So I would like to share today the work we have done on physical stimuli and the effect of wound healing. So the first of the 4 topics I want to discuss with you, is our exploration of mechanical stimuli. So we started with animal models, and we did an experiment where we compared in animal models affected by a model of rheumatoid arthritis. So this is a model for non-communicable diseases and chronic inflammation. And we treated these 2 arms with methotrexate, which is the gold standard pharmacological therapies, and a mechanical stimulation by needling in the back of the animals. We also compared this with a couple of arms of control arms, and finally also with a super control of where the animals were not induced for arthritis. And for all these animals, we also collected a variety of samples that we treated with omics. The idea was to have a sort of unbiased screen of what was happening. So we did the collection of these data at different time points. So before the beginning of any therapy, after 1 hour to assess whether there were early genes activated, and then at the end of the therapy, which lasted 34 days, to see the lasting effect of these stimulations. We did this in different type of tissues. So we did this in the subcutaneous tissue where the mechanical stimulation was released. And then to have a systemic assessment of what was ongoing, we also sampled blood and stools for analyzing the gut microbial composition. And finally, omics were done for transcriptomics and shotgun sequencing, for gut intestinal microbiome. So we had quite a lot of data with all these omics and all these data points. I want to highlight some of the most interesting, or at least some highlights. So first of all, of course, we could see that both the active arms, so methotrexate and the molecular stimulation, were able to reduce the thickness of the pores, which is the standard phenotypic parameters to assess the evolution of the disease of modern arthritis. And then we compared the expression, the gene expression across all the arms, of course, but here what we're interested in seeing is how this phenotypic aspect, so the decrease of inflammation and the reduction of the thickness of the pores, was mirrored at the molecular level. So here you see that there are functions that are shared in the two arms, where it's represented by these red lines that represent downregulated genes, mostly concerned with inflammation, and blue upregulated genes. Some of these are shared, but some are unique to the mechanical stimulation. And the function that is enriched, so the genes mostly belong to the function of wound healing. You certainly know, but let me briefly recollect that wound healing processes across 3 to 4 steps. So there's a very early non-transcriptional phase in the first hour after any stimulation or wounding. Then there is this inflammatory peak that was also discussed by previous speakers. And then we have the proper return to homeostasis via remodeling and regeneration. So we also checked the enrichment, so how the genes were changing by doing enrichment, so searching genes that belong to these different stages. And here you can see that the only process that go across all these phases is the stimulation, the mechanical stimulation. For methotrexate, this occurs in a completely different way, so the control of inflammation occurs through a different process, and nothing or a few things happen, of course, in the control arms. We then also checked what could be the systemic effect in the gut-intestinal microbiome. So one standard control is to check the diversity of the composition of the microbiome, and there is an increase in the diversity. And if we go deeper and check specifically which microbes were more abundant or less abundant with different therapies, we can highlight Roseburia and Lactobacillus, which are two bacteria that are known to have anti-inflammatory effect. In particular, Roseburia produces butyrate, which is a known anti-inflammatory metabolite. We also deepened a little bit this search to try to understand what was happening at the host microbiome interface. So we developed a relatively simple computational approach where we could somehow analyze together the genes of the host, so the rat, and the gene of the microbiome, and check whether together it was possible to see whether there was a shared function that they were enhancing or reducing. And indeed, we were able to observe that sphingolipid metabolism seems to be the function that is more affected at this interface. Then we did some translation in humans. So we have organized a pilot trial, a very small pilot trial. And here again, results, of course, need to be validated, in particular for statistical power. But what you can see here is a comparison between the two batches of experiments that we did in animal models. So batch 1 is the one I described before, and batch 2 is a study where animals were treated by mechanical stimulation alone or mechanical stimulation plus the therapy with methotrexate, which is also the therapy that the patients received. And again, you see that there is an upregulation of wound healing also in the human samples. So this wound healing function comes up again and again, and we thought that it was important to somehow have a tool that allow us to take this into consideration directly when we study inflammation. So we described with this, also in a collaboration also with Luigi Mani, who spoke just before me. And the idea is to consider not only inflammation and the innate immune response, and as Jean-François Moreau said, inflammation is everywhere, but not so easy to describe. It's too big, too ample. So we consider, we thought that it would make sense and would be helpful for our analysis to consider beyond the innate immune response, also the process involved in wound healing, Also, the process involved in the connection that there is between the autonomic nervous system and the control of inflammation, and also the host microbiome interface, because this also regulates, contributes to regulate inflammation. And while we did this, we published this sort of theoretical description, now we are trying to implement this. So basically, we would like to create in silico pathway where we can recollect and describe all these events and show their interconnection. So to do this, we collect the pathway that exists in public repositories. So Reactum is among the most renowned. We create the pathways that do not exist. So we compiled the in silico pathway for mechanotransduction because we couldn't find any. And now we are working at the optotransduction. In January, we start electrotransduction, and it would be great also to do magnetotransduction. And you see that the representation of this pathway with all these symbols, in fact, is a standardized representation, which allows to avoid as much as possible ambiguity. Every node is very clearly characterized, and so is every reaction. So we used a standard, which is called SBGN, System Biology Graphical Language, which is easily exportable, relatively easily checkable, and also enhanceable. And once we have these pathways, I want to show you some preliminary analysis we did. If we put together these pathways and we observe them as networks, and we compare this enlarged network, here we put together some of the pathways related to wound healing plus inflammation, we can analyze the relative position, so we say the topology of these nodes, how central they are, how they communicate with the rest of the network. And one type of analysis we can do is to check how the centrality changes. For example, in this preliminary data, we show that TNF-alpha changes a lot its centrality depending on how we interpret inflammation. Conversely, RAC, which is involved in the early stage of mechanotransduction, doesn't change so much. So it's a sort of, it has a sort of stable importance independently of what we are looking at, and so does NFKB. I talked about standardization, and we have discussed this also before. It is really difficult when working with non-pharmacological stimuli to have standardization, so to know the intensity of the stimuli that are appropriate to apply in a biological system. So recalling what Simona Villata presented this morning, I want to give you an overview of this project that we are doing in collaboration with the Polytechnic of Turin. So we developed this, they developed this 3D bioconstruct made of fibroblasts and keratinocytes in a matrix of collagen. So pretty simple, but this allows to study the effect in a 3D dimension, which, as we heard from Jean-François Moreau and also Carla Stecco, the three-dimensional component is really, really important. We extracted from those metabolites, RNA and DNA to study methylomics and transcriptomic, and we started to check what happens, what are the similarities and differences. So as Simona said earlier, we tried different type of electrical stimuli, and you can see, for example, that this is the up regulation and down regulation in samples that are stimulated at 5 volts with direct or alternate current. And it's very obvious that this elicits very different functions and type of response. So this is the type of work we would like to do to more and more stimuli to be able to compare apples with apples, that we say, while changing the parameters of the stimulation. And finally, another project that we did was to sort of address a curiosity and sometimes a frustration concerned with the way in which we can find publications about non-pharmacological stimuli. It's the literature is really scattered, and the narrative also about non-pharmacological stimuli it's somehow different and not always comparable with what happened for pharmacological research. So the idea to dig inside this difference, I thought that our computational methods were not sufficient, and we collaborated with anthropologists and a sociologist of science to try to understand what is the difference, or if there is a difference between the non-pharmacological treatment and pharmacological in the scientific narrative. So to do this, we selected a number of articles only from PubMed for the treatment of rheumatoid arthritis, simply because this is the model disease that I've used so far, searching therapies that can be referred to the greater inflammatory pathways. So we search for anti-inflammatory therapies, pharmacological anti-inflammatory therapies, which is the gold standard mainstream therapy, therapies that exploit the connection between the autonomic nervous system and in inflammation. So this is almost related to the vagus nerve stimulation. Therapies that affect the gut's intestinal microbiome, so mostly fecal transplant, and also therapy that affect wound healing, so different type of electrical, magnetic, optic, and mechanical stimuli. We kept for each of the stimuli 2 articles that were screened manually, and the idea was to read and score these articles across some social anthropological variables. Just to give you an example, one of these variables was temporality. The idea is to ask what is the reason, the rationale to promote a certain therapy. Is it rooted in the fact that it is based on an innovative discovery, and so this is innovation, or is it rooted in the fact that there is a long-dated practice, so this is based on experience? Or, for example, we have specialization, so this has to do with the fact that the disease is considered local, for example, to the joints, or systemic to the whole body. And then once we score this table, it's possible to apply very simple machine learning approaches, compact this information, and see that there are different representation of the therapies, pharmacological, experimental, so autonomic nervous system and gut-intestinal microbiome, and wound healing associated therapy that can be described in terms of these social anthropological variables. I will show you just one example of the type of results we get from this analysis. There are some more. So, for example, we saw that all the therapies that are related to wound healing somehow have their temporality in the experience. This means that, in general, authors do not consider all the work that we are doing, and they refer for therapy, for clinical applications, to tradition and habits more than to scientific discovery. This is just one example. The article is now on medRxiv. Hopefully soon it will be also published. So that's all I wanted to tell you, and thank also all the people that worked with me for that. If you have questions, we can discuss them. Or we have a few minutes if we want to see Carla's video. So we can put maybe, yeah, and you can try to share your screen.
I can try.
So present, share screen at the bottom.
Yes. Are you able to see now? Are you? You can see my screen?
Yes, and we see the movie.
Okay, so that is a contraction of the quadriceps muscle, and this is the fascia. I can show you again. I stop. And that is the deeper fascia, and you can see the layer white, that is the fibrous layer, and in the middle black is the loose connective tissue that is rich in hyaluronan. In this subject, you can see that when the muscle contracts, that layer move, and there is a changing in the loose connective tissue. if I show that other patient, you can see that there is not the same movement of the muscle, and above all, here, the different layer don't glide. They seems like attach one respect to the other, and the muscle are creating like a distortion of the fascia. So I don't know if the first problem is the fascia or is the muscle, but the two elements interact each other. And surely one of the key elements that we can visualize with the ultrasound is that lack of movement in between fascial layer. We know now that movement is allowed by the hyaluronan and the water, but we don't know the amount of, we know the amount of hyaluronan, but we don't know the molecular weight of the hyaluronan. And surely the characteristics of the hyaluronan change the viscosity of the loose connective tissue and can affect the gliding. I think that many physical therapy can modify the viscosity of the hyaluronan in that specific aspect, and consequently, we can come back from the second slide to the first slide.
Thank you very much. It was indeed very clear when watching the video. Luigi, if you can prepare yours later when we have a few more minutes, we will try to put yours. Now it's time to, so thank you, Carla. Now it's time to introduce Giancarlo Forte. He works at the St. Anne's University Hospital in Brno in the Czech Republic, and his work is mostly on cardiovascular diseases and mechanical stimuli. So he works in a hospital, so he's probably closer somehow to translation, and he can tell us more about his work. So, Giancarlo, please go on.
Hello, Christine. Can you hear me?
Yes.
And can you see my screen?
Yes.
Okay, so I'm definitely starting. So thanks a lot for the kind invitation. I'm not sure I'm closer to translation because, yeah, we do work in the hospital. We do, let's say, the first part of the translational work. So we work from the patient to from the bedside to the bench. But anyway, so I am the head of the Center for Translational Medicine at the International Clinical Research Center of St. Anne's University Hospital in Brno. And I lead the group which is called Mechanobiology of Diseases. So starting from the evidence that our cells are exposed to mechanical stimuli all the time, we work on the hypothesis that these functions of the apparatus cells use to perceive and respond to external mechanical cues contribute to pathologies. So we work with what is called the mechanosome. I know this is a very broad definition, but we basically start, and that's the translational part we entertain. We work with human samples and also with mice sometimes, but mostly with humans. And we start from the evidence that changes, the pathological remodeling of extracellular matrix is a big part of aging pathologies. We mostly work with the heart pathologies, and it's pretty obvious here how the change in extracellular matrix occurs during the pathology, where the muscle in red is substituted partly by a fibrotic tissue which is non-compliant and eventually will lead to the heart failure. So what we study in a nutshell is we try to address this question is how cells perceive pathological mechanical stress. And we have a very, very different definition from what I heard so far in the meeting of mechanosensing, or mechanosensor and mechanosensing. So we define a mechanosensor at the intracellular level as any intracellular component whose structure, localization, or function is modified as a response to changes in tissue mechanics and or nanostructure. And we mostly work on shuttling proteins. So I highlighted here a number of mechanosensitive entities in the cell, but we mostly work on shuttling proteins. So those proteins whose localization in the cell is altered by mechanical stress. And within the mechanosensitive proteins we study, one of the most famous, let's say the most successful so far, is a YAP-associated protein, which is the downstream effector together with TAZ of the Hippo pathway. The mechanosensitivity of Hippo pathway was established by Stefano Piccolo and Cyrio Dupont in 2011, showing that this pathway, which controls the shuttling of the two effectors I just mentioned to the nucleus, can be mechanically controlled, and it's sensitive to cell spreading, to stiffness, substrate stiffness, to stretching, and to flow. Basically the function of this pathway, which integrates biochemical and mechanical cues, is into controlling negatively the shuttling of these proteins by phosphorylation. So when the phosphorylation is removed, the proteins can actually travel to the nucleus, where they act as transcriptional co-activators in collaboration with context-specific transcription factors. And very obviously, you can follow. I'll be talking about the YAP mostly, but TAZ works pretty similar. The distribution of YAP in the cell can be pretty much followed, so that cell density controls its shuttling, but also substrate stiffness and cell spreading, as I mentioned before. And what is beautiful about this system is that the mechanical activation of the protein within the nucleus pretty much correlates with its transcriptional activity, and this can be impaired by inhibitors of intracellular attention. So what we demonstrated in 2017 was that when the protein is mechanically induced to shuttle to the nucleus of different cell types, it actually participates in the transcription of genes involved in focal adhesion assembly, so that cells depleted for the protein are not able to develop the classical focal adhesion spikes, which are deputed to interact with the extracellular matrix. And we also demonstrated that in a single cell setting, with cells being patterned being spotted on a micropatterned surface, cells losing YAP, they actually lose the ability to stretch and to develop a proper shape and size. And this led us to actually propose a model whereby YAP shuttling to the nucleus causes the activation of focal adhesion genes, and these strengthens cell interaction with the extracellular matrix. And this is very important for a number of different activities. We did this in breast cancer cells, but this interaction is key to the dissemination, for example, of metastasizing breast cancer cells. So we also demonstrated in another paper that the same protein is actually able to trigger the transcription of a handful of genes coding for proteins, for F-actin binding proteins, which contribute to cytoskeleton integrity. And in turn, we did this in pluripotent stem cells. We demonstrated that the cytoskeleton remodeling downstream of this mechanically activated protein contributes to the induction of mesoderm phenotype. And as a matter of fact, how this works, for a cell lacking the focal adhesions and the cytoskeleton integrity, it's a matter of force. So the cells are not able to get a grip on the extracellular matrix, as shown by traction force microscopy. And again, when the protein is depleted, the focal adhesions are not formed, so the cells are not able to produce a contractile force, not inward, not outward. And so what about the heart? Because I said we work mostly with the heart. So this protein is very important in cardiogenesis, and that's the important part. So that's a mechanically activated protein which works which triggers the proliferation of embryonic cardiomyocytes. So when the protein is depleted, the heart simply doesn't form, and the embryo is lethal. And it works in regeneration. There's a tiny phase of regenerative heart postnatally. And then the expression of this protein is abolished in the adult heart unless it gets reactivated when a mechanical stress is applied, like a myocardial infarction, but its sustained activation leads to disease, to dilated cardiomyopathy, to hypertrophy. And most importantly, the group of Marojaka recently demonstrated that in the adult heart, pig heart, YAP is downstream of a network of microRNAs which are able to trigger the reactivation of proliferation and heart regeneration. So what we did, we approached the issue of pathological mechanosensing in the heart from the other side, from the extracellular matrix. So we were able to get our hands on a number of different human hearts from transplanted patients, and we decellularized them by stripping away all the cells. We were left with the extracellular matrix, which we actually imaged under a 2-photon microscope, and obtained a computational fluid dynamics at a very high resolution model of ECM pathological remodeling. And by this, we got a fingerprint of how ECM remodeling occurs in the pathological heart. And also, we were able to correlate the clear reduction in heart functionality in these patients, as measured by ejection fraction, with a similar reduction in the complexity of the ECM, as measured by second harmonic generation. And to this was associated as a similar reduction in the compliance of the extracellular matrix of the human heart. And what we demonstrated was that the 3D rearrangement of the extracellular matrix is indeed sufficient to activate YAP TAD transcriptional activity in cardiac fibroblasts, and this leads to an increased contraction of the cardiac fibroblasts, which eventually are more able to digest the extracellular matrix, to remodel the extracellular matrix, and to actually worsen in a feedback loop the pathology. So also in 2014, we demonstrated that the reactivation of YAP in pathological heart also occurs in cardiomyocytes, not only in cardiac fibroblasts. And in order to study what this means for the cardiomyocytes, we actually resorted to pluripotent stem cells. We obtained contracting cardiomyocytes, which are able to handle the calcium fluxes, and we established a model of volume overload by static uniaxial stretching, and we demonstrated that this mechanical stress is indeed able to trigger YAP relocation to the nucleus of these cardiomyocytes. Then what is the result of YAP reactivation? This was the next question, and we actually demonstrated that the reactivation of YAP affects deeply cardiomyocyte function by changing the beating frequency, the activation potential, by changing the calcium extrusion, and eventually affecting the pacemaker currents. And then we also found, by using a model of familiar dilated cardiomyopathy obtained from a patient having a single mutation in cardiotropin B, we showed that these patients, they have a reduced contractility of the cardiomyocytes, but when you re-express YAP in these cells, you can actually restore the contractility to levels similar to the healthy counterpart. Another interesting study we did recently and was just accepted for publication regards the mechanosensitive RNA metabolism in pathological heart. So altered RNA metabolism in the diseased heart is a well-known phenomenon occurring, which leads to the aberrant alternative splicing of a number of transcripts involved in cardiovascular function. And a few years ago, we were able to demonstrate that one protein, which is deeply involved in the splicing of of a number of these transcripts is reproducibly upregulated and displaced in the pathological heart. We demonstrated this in the mouse heart, in the human heart, in chronic conditions, in acute conditions. So invariably, this protein, which is called HNRMPC, heterogeneous nuclear ribonucleoprotein C, shuttles in pathological heart from the nucleus to the sarcomeres, so outside of the nucleus. So we wonder whether the splicing would be affected and whether this is connected with the ECM pathological remodeling and the mechanical stress associated to the phenomenon. So we put our hands on what we call the mechanobiology toolbox to study the possible mechanosensitivity of this protein. And so we studied patient specimens. We produced iPS-derived cardiomyocytes. We did 2D micropatterning, 3D encapsulation, and also we used a lot of bioinformatics and molecular biology to answer this question. And eventually we demonstrated in 3D hydrogels that the localization and then the function subsequently of the splicing factor depends pretty much on cell 3D spreading. In 2D, by using micropatterned islands for single cell entrapment, we demonstrated that the localization is still affected by cell size, and this depends on intracellular tension. Therefore, we demonstrated that this protein, the localization of this protein, is in fact mechanosensitive. And then we depleted the protein with two different approaches, genetic approaches, and demonstrated that the absence of the protein from the nucleus of cardiomyocytes affects deeply the alternative splicing of more than 2,000 events. We found more than 2,000 events, and among them we found an annotation for the Hippo pathway, which I was discussing before, and for YAP target. And eventually we showed that the splicing factor is needed under physiological conditions in the nucleus of cardiomyocytes for the splicing of exon 4, which encodes for the WW domain in the YAP messenger RNA. And when the protein is not there, this splicing is affected, so that, as we demonstrated in another story published last year, this aberrant splicing, it's in fact part of the pathology somehow, so that the isoforms are spliced differentially in the healthy and the pathological human heart. Thereby, we described in this case a new layer of mechanosensing, which is driven by the regulation of alternative splicing in the diseased heart. We also demonstrated at the time that the mechanical impact of pathological ECM on the splicing occurs through the phosphorylation of protein kinase C, which drives the shuttling of the splicing factor outside of the nucleus, thereby affecting the alternative splicing of a number of messengers, as I showed before. And with this, I'd like to thank you for the attention, and I'd be happy to take any questions. Thanks a lot.
Thank you very much for this careful presentation of all the effects of mechanical stimuli on the heart. So there is a question that you partially addressed from Vitaly Zablotsky. How did you apply mechanical forces to cell? You've shown this device. Maybe you want to give a little more information about it.
Yeah, this is it's a mechanical stretcher from a company called Cell Scale, which can apply uniaxial, either static or dynamic stretching, and that's how we did it. We did it static. We also do it dynamically, but in this case, it was a dynamic stretch. You basically see the cells, and then you stretch them and leave them under tension for 24 to 48 hours.
Okay, thank you. More question? Up.
Yes, I would like to ask, okay, you stretch all the big amount of cells, it's interconnected, but how do you know the force will you apply it to individual cell and even for individual protein? How big is this force? I suppose
no, these are individual cells. These are individual cells. So you can have like two different regimens. Yeah. So you can have two different regimens. with cardiomyocytes. Cardiomyocytes are syncytium, right? So they are very well connected. So you can actually obtain a monolayer of cells and stretch them from side to side. On the other hand, what we did here, we obtained single cardiomyocytes. We seeded them single on the extracellular matrix, and we stretched the extracellular matrix. Okay,
Okay.
that's the best we
Thank you.
can do.
Thank you.
Thank you. One more question from Jean-François Moreau. Cycles of contraction also create pressures on cells. What about pressure instead of traction? Is there dedicated pathways to sense pressure?
Yeah, there are. I think there are, but we never did this. So there is a tendency to actually consider stretching and pressure as not antithetic, but very different stimuli somehow. But pressure is felt is perceived through mechanosensitive channels, right? So the TRPV4, for example, and here we are talking about the protein, which is somehow indirectly induced to shuttle with any kind of mechanical stress. So it's a very sensitive protein. It's like a shock protein.
Thank you very much. Thank
Thanks.
you. So we can move now to the last speaker. This is Timothy Ko from the Department of Kinesiology and Nutrition in the University of Illinois, Chicago. Welcome. He's interested in mechanisms that regulate tissue inflammation, and he will tell us something about yet another type of mechanical stimulation, which is low intensity vibration.
Thank you.
Welcome, Timothy.
Can you hear me?
Yes, we
Good,
do, and
great.
we also see your slides.
Great. Okay. Thank you. And thanks for the invitation to present here. Sorry, I'm seeing my lights flashing. Let me turn that off. Seems to be maybe my camera. Hopefully that won't be too disconcerting. So my lab studies the role of inflammation in tissue repair, including the role of low intensity vibrations in both skin and skeletal muscle. We are interested in developing methods to improve healing of both skin and skeletal muscle using biological and mechanical approaches, and a little bit of work with cell therapies too. For this talk, we'll focus on the effects of low intensity vibrations. Going back, looking historically, the first mention that I've seen of people using vibrations to affect the human body came from Nikola Tesla, who apparently was interested in the effects of vibrations on various systems in the human body, including the stomach, the kidney, the heart, and so on. There's an anecdote that he was treating people with mechanical therapy, including Mark Twain, who was apparently suffering from some maladies and was apparently cured by this by these mechanical stimuli. Lately, more recently, people have been using vibration therapies in to try to improve health in a number of different ways, including bone, so trying to prevent bone loss, improving bone healing. There's a number of studies looking at strength training of muscle and improving muscle repair, improving rehabilitation in various neurological disorders, reducing impact of high-fat diet on the development of different aspects of diabetes, including improving insulin resistance, and as well, what I'm going to talk about today, improving wound healing, including pressure ulcers and diabetic skin wounds. Chronic wounds are a significant problem, not only here in the US, but around the world. The causes of healing, some things are known, but we still have a lot to learn. And we and a couple other groups have found that low intensity vibration increases skin blood flow, and in humans anyway, there is some work done on pressure ulcers. Our work that I'm going to present today is in mice, and we're trying to get ready for some studies in humans. So we've done work both with whole body vibration, shown there on the left, where we have a vibrating platform. We put the mice in a cage on top of the platform, and we deliver vibrations to the whole body in that manner. We are also starting to do local vibration application to, in this case, anesthetized mice. And this is effectuated by a little shaker that's applied to a spot on the back of the mice. The data that I'm going to show you is the effects of whole body vibration on diabetic mice. Wound healing, skin wound healing in diabetic mice is impaired. You see delayed wound closure, severely impaired formation of granulation tissue, impaired angiogenesis, and this is all associated with a chronic inflammatory response. And what we've seen is in this slide that application of a whole body vibration with a specific protocol improves healing of the wound, the skin wounds that are made on the back of the mice with an 8mm biopsy punch. So on the left, histological image shows what the healing response of control mice. So control mice were sham treated, placed in that cage on the platform without vibrations. And on the right, you see the healing response in mice that were exposed to the vibration protocol. And we see that what we're calling our kind of low intensity protocol showed increased or accelerated wound closure, so growth of the epithelium over the wound, whereas protocols with higher vibration intensity did not have a positive effect, and in some cases at the highest intensity showed impaired wound healing even compared to the controls. Probably the most significant effect that we're seeing with the effects of these vibration protocols is on the dermal healing, the granulation tissue formation, and angiogenesis. And again, this low intensity protocol showed accelerated or enhanced granulation tissue formation associated with enhanced angiogenesis, and it's marked in that center graph by CD31 staining, and that's what's shown above the histological images showing the enhanced angiogenic response with vibration there on the right. We also see that the vibration protocol reduces kind of the chronic inflammatory response in the wounds. So here we're looking at neutrophil accumulation of the wounds. Typically, the early response is not affected tremendously, but the characteristic chronic inflammation, the inflammation that doesn't resolve in these wounds, is ameliorated by the vibration protocol. Here again, shown in the neutrophil response. we're starting to look at kind of the mechanisms that are involved here. We started to take a look at growth factors that might be changed by the vibration protocols in the wounds. And what we're seeing consistently is that levels of IGF-1 are increased in the wounds by the vibration protocols that improve healing. We also tend to see increased VEGF levels in the wound, whereas other growth factors like FGF and PDGF have not been affected. And we've looked at a number of different growth factors that also are not affected. So to look at the IGF-1 story in more detail, we kind of wanted to see if this was a local response or a systemic response. IGF-1 is an interesting growth factor because it circulates at relatively high levels and the systemic levels of IGF-1 are predominantly produced by the liver. So we looked at IGF-1 levels in the wound, in the blood, and in the liver. And we found that IGF-1 levels were increased in all those tissues by the vibration protocol, with a little bit different time course, but increased at different time points. We also looked at IGF-1 message levels in the wound and in the liver and found that early on, vibration increased IGF-1 message levels in the liver, and at a time when IGF-1 protein levels were also increased in the wound, and IGF-1 message levels in the wound were increased, but only at a later time point. So this kind of begged the question of whether the increased levels of IGF-1 in the wound induced by vibration were a local or a systemic response. So we went on to develop or use a model where we could knock down IGF-1 specifically in the liver to see if that influenced our vibration-induced response in the wound. So knockdown of liver IGF-1 decreases circulating IGF-1 by like 90%. You can see that in the center graph. It's very effective and very specific to the liver. And knocking down IGF-1 in the liver also reduced IGF-1 levels in the wound, which suggests that liver IGF-1 is responsible for a lot of the IGF-1 that we see that accumulates in the wound. And under those conditions, the improved wound healing we see with vibration in the wound is abrogated by this liver IGF-1 knockdown, suggesting that the liver IGF-1 is induced by vibration, may be playing an important role in the healing response that we're seeing. So when we look at the healing response, we see similar trends. So the improved healing we see with vibration is abrogated when we knock down the liver IGF-1, both in terms here of wound closure and in terms of the dermal healing response. The granulation tissue that's increased by vibration does not improve when we knock down liver IGF-1. And same thing with the angiogenic response that's induced by vibration is not induced when we knock down liver IGF-1. So this is a little bit of the data that we've generated on this project. We're showing that low intensity vibration enhances angiogenesis and granulation tissue formation in diabetic mice. We've done this both in genetically diabetic DB mice as well as in high-fat diet-fed mice. It also accelerates wound closure in these mice. We're seeing an effect that is maybe dependent on IGF-1, at least that's playing a role. And we're seeing that there is an anti-inflammatory effect associated with this, and that the liver, there may be a systemic response to the vibrations we are applying that involves the liver and communication between the liver and the wound. And with that, just thank the people that actually did the work here. Rita Mirza is a longtime person in my lab that's done a lot of work throughout the years. Eileen is a postdoc who helped publish the first paper, and then the clinical collaborators that we're working with now to try to translate this work to a human study. So thank you, and happy to take questions.
Thank you very much. There are indeed questions for you. So
Okay.
The first from Jean-François Moreau is there a correlation between neutrophils infiltration and edema under the vibration protocol?
That's a good question. We haven't really addressed edema in the wound, so I can't answer that question. I don't see any, frank evidence of edema, but we haven't really followed that up or focused on that.
Okay. Yeah, edema should ease the cell circulation. That's the comment of Jean-François.
Yeah, there's been interest in. I've had a number of people contact me about whether we're seeing any change in lymphatics, and we just haven't gone there yet. So, and then
sorry. Yeah, I put you on stream to have an easier conversation.
Okay.
But I think he's satisfied.
Okay. And then the amplitude of the vibration. So the amplitudes we're using are very small. They're less than 0.1 millimeter. We are controlling the accelerations between so low intensity vibration is typically defined as anything below 1G acceleration. And so we varied between 0.3 and 1G, and we varied the frequencies between 30 and 90 hertz. And it seems like the parameters on the low end are the ones that are improving the healing response. Part of it is an issue with the equipment, and the higher amplitudes tend to make a lot of noise, and so we're trying to avoid that especially because these mice are awake, the stress on the mice and the stress on the investigators. So we again, when you look in the literature, there's a wide variety. I think Christine was kind of pointing this out in different modalities, that there's a wide variety of parameters that people are using, including displacements 3-4 millimeters down to what we're using, and accelerations of many multiples of G's. Some of the effects are similar. Skin blood flow tends to increase with almost any vibration, well, up to a certain limit. But I was kind of surprised that the healing response was only positive healing response was only affected by those lower intensity protocols. I've had conversations with a colleague of mine who looks at the effects of these vibrations in bone, and they don't see such a strong parameter dependency. But again, it's a pretty limited range that they've been testing. So.
Thank you. This goes back again to standardization
Yes.
and the need to understand more about the
Definitely.
intensity of the stimuli. If
Definitely.
there's no further questions, we can stop broadcasting and go into the private discussion, the second part of the private discussion. Okay, no more questions. So I think we can close the broadcast. Thank you everyone who participates, speakers and listener outside. Thank you.